Hello everyone, welcome to IEEE Sensor Conference 2021. I, Shradha Suman Panda, am here to present the research work on title An Approach Towards Development of Point-of-Care Diagnostics Using ELISA. This work has been carried out as a collaborative effort between IIT Delhi and IIT Jodhpur. The outline of my presentation is as follows, starting with a general introduction, followed by objectives, experimental detail, results and discussion, work progress strategy, conclusion, and references. Starting with general introduction. Diagnostic technology has advanced substantially over the last few years, and recent years have witnessed tremendous advances in point-of-care diagnostics, which are a result of continuous developments in biosensor, microfluidics, bioanalytical platforms, assay formats, lab-on-a-chip technologies, and other complementary technologies. The growing demand of point-of-care diagnostic in healthcare sector is not only helping the doctors, but also the patients to make better clinical decisions and provide cost-effective treatment to them. Point-of-care diagnostics are further primarily designed to acquire clinical information from patients, especially in resource-limited settings. Some of the greatest benefits of employing these testing systems involve the rapid result interpretation for clinical decision making, with an additional perk of achieving real-time results similar to lab quality diagnostic results in lesser time span. Biosensor is one of the most critical component of this point-of-care diagnostics, and it is directly responsible for bioanalytical performance of an assay. And ELISA-based methods are well known for their biosensing applications. The figure on the right-hand side depicts a mobile ELISA device as a point-of-care diagnostic. Some of the key objectives of our research work is as follows. To explore the unique features of nanostructured silicon as a substrate for ELISA testing, quantitative and qualitative analysis of analytes and their concentration present in sample. Further, establishing a correlation between optical density obtained with the help of ELISA reader and mean RGB value obtained with the help of image processing technique in order to develop a point-of-care diagnostic applicable for mobile health applications. Talking about the experimental details, I would like to start by defining some steps that we have utilized while fabricating the sensor. At first, the silicon wafer was prepared with specifications as mentioned in the first block, followed by porous silicon sample preparation using an electrochemical etching process with the silicon wafer. The wafer was further etched utilizing an in-house Teflon cell at 20 milliampere per centimeter square current density for 20 minutes in hydrofluoric acid and the ethanol solution as an electrolyte. This was also optimized at different current densities and etching duration. And all the etched wafers were further dried in nitrogen and cut into small pieces of 8 millimeter into 5 millimeter and further proceeded for biosensing testing. The figure on right hand side demonstrates an in-house made setup for electrochemical etching of silicon. Some of the different types of substrates that were utilized in our research work are tabulated as follows. Starting with porous silicon, followed by aptis functionalized porous silicon, crystalline silicon, aptis functionalized crystalline silicon. And the negative controls of all these samples were also tested. The functionalization of the sample. The biofunctionalized substrate, as seen in this figure, was mostly prepared and incubated for around 1 hour 15 minutes at 37 degrees Celsius in GAR, which is used as a primary antibody. Goat antirabbit IgG act as a primary antibody as shown in the figure. Rabbit antigoat IgG is acting as an analyte, and the HRP conjugated is used as a secondary antibody. Further, various steps were utilized for functionalizing these samples, starting with the silicon wafer and porous silicon wafer. They were cut into small pieces, and all these pieces were further functionalized utilizing 10% solution of aptis in toluene at room temperature for around 1 day. and this toluene will further reduce the probability of condensation of Aptis molecule within the solution. A sandwich format ELISA testing was performed on all the substrates that were mentioned in the previous slides to see the capabilities of those substrates. The characterization of samples. In the first figure, it represents the FTIR for comparing two substrates and demonstrate the nanostructured porous silicon before and after the attachment of Aptis. And it was observed that after the attachment of Aptis on the porous silicon, we can identify the presence of amine groups as shown in figure. And these amine groups are important for attachment of GAR on their surface. The second figure depicts a Raman spectrum for comparing nanostructured silicon and crystalline silicon. From the Raman spectrum, it was observed that in case in which functionalization of the Aptis was done on nanostructured silicon, there were peaks that were more intense and which could lead to further attachment of more GAR on the surface. And these peaks were further obtained for NH2, that is amine, and CH2 groups. And hence, less attachment was observed on only silicon with Aptis. Followed by the work progress slide. As shown on the right hand side, the two figures represents the ELISA wells. The optical density was first measured with the help of an ELISA reader and picture of the samples were taken for image processing. Image processing was conducted with the help of MATLAB software. Further, we estimated the mean RGB value and then we plotted a graph between mean RGB versus substrate and also mean OD value with different substrates. And um, we observed that both the OD and the RGB index they found to be increasing with the increasing order of RAG. And further, we planned to develop an app for the diagnostic application. And currently, we are working on cancer antibodies to produce a point of care diagnostic solutions. Mean RGB, mean OD versus the substrate concentration. The first figure represents the optical density along with that of the substrates, and the second graph represents the mean RGB value along with different substrates. From the first graph, it was concluded that the mean OD was found to be maximum for APSI, that is the Aptis functionalized porous silicon, and it was mainly due to the availability of Aptis molecule to react with wafer. The surface area will increase, which further results in more effective salinization of wafer surface. Both the optical density and the RGB index were found to increase with increasing order of RAG. And it was also possible to correlate the value of optical density obtained from the ELISA reader and the RGB index value obtained with the help of image processing technique. And this image processing techniques is further planned to be utilized for developing an app for diagnostic solutions. The different sensing mechanisms. Here, the figure on the right hand side basically shows the formation of a siloxane network between the Aptis molecule and the porous silicon surface. When the silicon or the porous silicon substrates were incubated in Aptis solution, the hydroxyl group which are present at the oxidized porous silicon will lead to chemical modification of ethoxy groups of Aptis and further will result in the formation of a siloxane network. Conclusion and future work. ELISA-based methods are well known for biosensing application. This work presented nanostructured silicon as a sensitive substrate for attachment of GAR and RAG model antigen and antibodies on the surface. Further, it was possible to correlate the value of optical density and RGB index obtained from the image processing technique. Best results were further obtained from the salinized porous silicon sample due to the presence of nanostructures. The study is being extended for development of point-of-care diagnostic solutions for detection of cancer biomarkers. And we are mostly planning to prepare a point-of-care diagnostic solutions for it. These are the list of references that I have referred to while preparing this research work. That's all. Thank you.